A cancer vaccine has passed phase 3. That is a real milestone. For patients whose high-risk melanoma was removed, the idea is deeply personal. Doctors take that patient's tumor. They identify its mutations. Then they manufacture a treatment designed to help stop invisible cancer cells from returning. But the headline hides two crucial facts. The vaccine was tested with Keytruda, not by itself. And we still do not know how large the phase 3 benefit was. So this is the real story, a genuine first, with the decisive numbers still missing. The therapy is called intismeran autogene, previously V940. Doctors begin with the patient's removed melanoma. They sequence it and compare its mutations with healthy cells. Software identifies abnormal proteins, neoantigens, that could expose remaining cancer cells to the immune system. Up to 34 targets are encoded into a synthetic mRNA recipe made for that one patient. After injection, cells read the recipe and display those targets. T cells can then learn the tumor's molecular fingerprint. This is a treatment vaccine, given after cancer has already developed. It is not a preventive shot for the general population. The vaccine is paired with pembrolizumab, sold as Keytruda. It blocks PD-1, a checkpoint tumors can exploit to suppress T cells. The simplest analogy is a custom-wanted poster plus a release break. The vaccine identifies the target. Keytruda helps the immune response proceed. So the wrong comparison is vaccine versus nothing. The actual question was whether adding the custom vaccine worked better than Keytruda plus placebo after surgery. This result belongs to the combination. It does not establish vaccine monotherapy. Interpath 001 enrolled 1,137 people with high-risk stage 2B through stage 4 cutaneous melanoma after complete resection. They were assigned 2-to-1, under double-blind conditions, to the combination or the control. The first endpoint was recurrence-free survival, how long patients remained alive without the melanoma returning. The second was distant-metastasis-free-survival, how long before it spread elsewhere. The combination crossed the trial's statistical bar on both. The companies call the improvement clinically meaningful and report no new safety signal. That makes this the first positive phase 3 readout for an individualized neoantigen therapy and for an mRNA-based cancer treatment. Here's the break on the celebration. This is still a top-line announcement. We still do not have the phase 3 hazard ratios, absolute recurrence rates, confidence intervals, subgroup results, or detailed safety table. Overall survival, whether patients live longer, is still being followed. You may see an attention-grabbing number, 49% lower risk of recurrence or death. That came from the earlier phase 2 study at roughly 5 years. It is useful history, not the new phase 3 effect size. The earlier randomized trial had only 157 participants. Phase 3 expanded that test more than 7-fold. Now the complete results need presentation and peer review. If the full result holds up, the larger breakthrough is a repeatable oncology pipeline. Remove a tumor, sequence it, select targets, manufacture custom mRNA, and combine it with checkpoint therapy. Moderna and Merck are testing the platform across lung, bladder, kidney, and other cancers. But a platform is not a universal cure. Tumors and immune systems differ. Manufacturing takes time. And success after surgery in high-risk melanoma cannot be assumed in every cancer or disease stage. Regulators and doctors will judge the complete benefit and risk, not the novelty of the recipe. The bottom line is bigger than hype and narrower than a cure. A therapy built from one person's tumor has passed its first large late-stage test. The next evidence is the size of the benefit, the safety detail, and whether patients live longer. The frontier explained in 5 minutes.